Hi student, today we all will be learning about the glycogenesis. So glycogenesis is the synthesis of glycogen from glucose is glycogenesis. Glycogenesis takes place in the cytosol and require ATP and UTP beside glucose. The first step is synthesis of UDP glucose. The enzyme hexokinase and glucokinase convert glucose to glucose 6-phosphate. Phosphoglucomutase catalyzes the conversion of glucose 6-phosphate to glucose 1-phosphate. Uridine diphosphate glucose is synthesized from glucose 1-phosphate and UTP by UTP glucose phosphorylase. Then comes the second step, which is the requirement of primer to initiate glycogenesis. A small fragment of pre-existing glycogen must act as a primer to initiate glycogen synthesis. In the absence of glycogen primer, a specific protein, namely glycogenin, can accept glucose from UDPG. The hydroxyl group of the amino acid tyrosine of glycogenin is the site at which the initial glucose unit is attached. The enzyme glycogen initiator synthase transfers the first molecule of glucose to glycogenin. Then glycogen itself takes up a few glucose residue to form a fragment of primer which serves as an acceptor for the rest of the glucose molecule. The third step is glycogen synthesis by glycogen synthase. Glycogen synthase is responsible for the formation of 1,4-glycosidic linkages. This enzyme transfers the glucose from UDP to glucose to the non-reducing end of the glycogen to form alpha 1,4 linkages. The fourth reaction is the formation of branches in glycogen. Glycogen synthase catalyzes the synthesis of a linear unbranched molecule with 1,4 alpha glycosidic linkages, which includes the formation of branches is brought about by the action of branching enzyme namely glycosyl alpha 4,6 transferase. This enzyme transfers a small fragment of 5,2-A glucose residues from the non-reducing end of glycogen residue from the non-reducing end of glycogen chain to another glucose residue where it is linked by alpha 1,6 bond. This leads to the formation of a new non-reducing end besides the existing one. Glycogen is further elongated and unbranched respectively by the enzyme glycogen synthase and glycosyl 4,6 transferase. The overall reaction of the glycogen synthesis for the addition of each glucose residue is glucose N plus glucose 2 ATP, which gives glucose N plus 1 2 ADP plus pyrophosphate. Of the two ATP utilized, one is required for the phosphorylation of glucose, while the other is needed to conversion of UDP to UTP. Here comes the glycogenesis reaction. Glucose converted into glucose 6-phosphate glucose by glucokinase. Then it converted into glucose 1-phosphate by phosphoglucomutase. Then this changes into UDP glucose by the enzyme UDP glucose phosphorylase. Then it changes into glycogen initiator synthase and changes into glycogen primer. It changes to glycogen synthase to alpha 116 4,6 transferase, then it go to alpha 1,6, then elongation by glycogen synthesis, branching glycosyl 4,6 transferase, it forms glycogen. This is glucose 6 phosphate, glucose 1 phosphate, UDP glucose. The show whole of the reaction branching done and form with glycogen. Thank you.